Welcome to the deep dive. Today we're looking at something pretty complex but also incredibly important. Inherited retinal degenerative diseases, or RDDs.
Uh-huh.
These conditions, while they systematically attack photoreceptors, the light-sensing cells in our eyes, and often the end result is severe vision loss, sometimes complete blindness.
It's a devastating group of diseases. So our mission today is really to get up to speed quickly on how human retinal organoids are changing things.
Right. These organoids built from induced pluripotent stem cells, iPSCs.
Exactly. We need to explore how these iPSCs aren't just, interesting lab curiosities. They actually let us model human retinal development in a dish.
And model specific diseases, too.
Yes, things like gyrate atrophy, retinitis pigmentosa,
Yeah.
and even test potential therapies in a really personalized way. It's a fundamental shift.
So we're talking about building a functioning 3D model of the human retina, not just growing flat layers of cells.
Precisely. And it all hinges on pluripotency. These iPSCs, which remember can come from just a small skin or blood sample, they have this incredible ability to become almost any cell type in the body.
Which makes them perfect for looking at early development, stages we couldn't really access before.
Absolutely. And for RDDs, it's particularly powerful because often these diseases start out by hitting just one specific type of retinal cell first. Makes them easier to study in a controlled, structured model like an organoid.
Okay, so let's get into how you actually build one. How do researchers take these, very versatile stem cells and coax them into forming something as intricate as a retina?
It's fascinating, actually. It's highly orchestrated, but a lot of it involves letting the cells follow their own sort of internal developmental program. It mimics the timing found in nature pretty closely.
So what does that look like in the dish?
Well, the initial cultures, they start as general stem cells.
Right.
And they progress to form structures that look like the early optic vesicle.
Okay.
And then, this is the key bit, they form these three-dimensional retinal cups, or RCs.
And these RCs, they just spontaneously sort themselves out into the different parts needed for vision?
They really do.
Yeah.
It's quite remarkable. You see progenitor cells that initially express both VSX2 and MITF.
Right.
These are key developmental markers.
Right.
But then, just like in normal development, they segregate. The VSX2 positive cells form the part that will become the neural retina.
Mm-hmm.
And the MITF positive cells form the retinal pigment epithelium, the RPE layer. Perfect segregation.
And once you have that basic separation, the layers start to build up.
Exactly right. The 3D RCs mature, and eventually you find all the major players. Ganglion cells, photoreceptors, amacrine cells, horizontal cells, Mueller glia. They're all there.
And in the right order of appearance.
Spot on. The timing is correct. Retinal ganglion cells, the RGCs, which send signals to the brain, they appear first. Then later, the photoreceptors, the light catchers, differentiate. It follows the natural blueprint.
This sounds amazing, but also maybe really fiddly and time-consuming. Like, if every lab is manually handling these tiny structures, that must slow things down.
Oh, it absolutely did. Traditionally, isolating these little retinal structures involved very careful manual microdissection. Could take, 40 minutes or more for a batch. A real bottleneck.
But I remember reading one of the sources mentioned a big improvement there, something about scraping?
Yes. A major practical advance. They developed this checkerboard scraping technique. Sounds a bit rough, maybe.
A little, yeah.
But it works. It basically allows them to quickly detach and collect the desired structures.
And how quick is quick?
It brought the isolation time down from 40 minutes to about 5 minutes. Just 5 minutes.
Wow, okay. That's huge.
It is. And combined with other improvements, like using high-density microwell arrays for seeding the cells, they could generate something like 3,700 organoids per plate. You take maybe 700 or so.
Okay, 3,700. That's not bespoke anymore. That's practically industrial scale for this kind of research.
Exactly. That jump in yield, it really changes the game for things like drug screening, because it's actually feasible on a larger scale.
Right. It moves from being a cool possibility to something genuinely scalable for testing lots of potential drugs.
Absolutely. Hundreds of tests in parallel become possible.
Okay, so we've got the structure and the scale. Let's talk function.
Right.
RGCs, the ganglion cells, they're the retina's connection to the brain. Any cell replacement therapy needs these neurons to grow axons, sometimes over long distances, to reconnect properly. Can the organoid model test things that help that happen?
It can, yes, and that's a really critical application. First off, the RGCs arrange themselves correctly in the organoid in the basal layers where they should be.
Okay.
So using this setup, researchers tested factors that might influence their outgrowth, their neurites. They found the substrate they grow on matters laminin worked very well.
Uh-huh.
But the really interesting finding was a soluble factor, something called netrin-1.
Netrin-1, what effect did that have?
It significantly boosted both the average length of the RGC neurites and how many neurites grew out. Some of these extensions reached almost 1.5 millimeters in just 24 hours.
1.5 millimeters in a day. That's serious growth.
It really is. And if you think about therapies like cell transplantation, understanding what encourages that kind of rapid directed growth is absolutely crucial for making connections.
Yeah, that feels like direct evidence the system can model active wiring, or the beginning of it. And that growth is driven by the very tips of these neurites, isn't it?
That's right, the growth cones. These are dynamic structures at the leading edge, constantly sensing the environment. They found these growth cones were rich in F-actin, very motile, and showed increased forward movement when they were exposed to that netrin-1. So the organoid lets you see that mechanism in action.
Okay, let's shift focus to the photoreceptors themselves, the rods and cones. How mature did they get in these organoids? And crucially, could they actually detect light?
Structurally, they got quite far. They developed inner segments, connecting cilia. These are important internal parts. They even started forming outer segments, or OS.
That's the part that actually captures the photons, right?
Exactly. But, and this is an important caveat, the really mature structures inside the OS, the stacked membrane discs, they were only seen at a low frequency. So not fully mature like in an adult eye.
Okay, so there's a structural limitation there, probably due to the in vitro environment. What does that imply? Can we still trust the functional results?
It's a fair question. It means we need to be a bit careful drawing conclusions that depend only on fully mature outer segments. However, the system was definitely functionally responsive.
How did they test that?
Using electrophysiology. They recorded electrical signals and confirmed that the photoreceptors themselves did react to light flashes.
Okay, so the basic machinery is working.
Yes. And even more convincingly, when they recorded from the ganglion cell of downstream neurons, they found that 82% of them showed significant responses when light stimuli were presented.
82%. So even with somewhat immature photoreceptors structurally, the message is getting through. The synapses are working.
Precisely. Functional synaptic connections, those specialized ribbon synapses, were clearly present and transmitting the light signal down the chain. That's the critical proof of concept needed before you can really trust it for disease modeling.
We have a working human circuit, albeit maybe not fully adult-like. That's the key takeaway.
That's it, a functional baseline.
All right, let's pivot to the really personalized applications. Tell us about the work using organoids to model gyrate atrophy, GA.
Okay, so GA is an RDD that mainly hits the RPE, the support layer behind the photoreceptors. It's caused by mutations in a gene for an enzyme called OAT.
OAT, okay.
Researchers took cells from a patient with GA, made iPSCs, and then differentiated them into RPE cells. And sure enough, these patient-derived RPE cells showed the expected problem. Very, very low OAT enzyme activity. A real disease-in-a-dish model specific to that person.
And the first thing they tried was using it for drug testing, right? Looking at vitamin B6.
Exactly. It's known that some GA patients respond to high-dose vitamin B6, so they tested different concentrations on these patient-specific RPE cells.
And what did they find?
They found that a concentration of 600 micromolar 600 Cre dramatically boosted the OAT activity, like a 24.5-fold increase. It really worked for this patient-specific mutation.
So why is it so important to nail down that specific dose, 600-fold? I mean, it's just a vitamin, isn't it?
Well, yes, but too much can be harmful. This screening also showed that a lower dose, 200 OM, didn't really work. And crucially, going higher than 600 OM gave no extra benefit.
Ah, I see.
And excess B6 isn't harmless. It can cause nasty neurological side effects, sometimes permanent nerve damage. So this organoid model allowed them to find the optimal effective dose for this specific patient before treatment. Maximum benefit, minimum risk, precision medicine in action.
That's a powerful demonstration. And they didn't stop at drugs, right? They also tested gene correction.
They did. They used a technique involving something called a bacterial artificial chromosome, or BAC.
Okay, BAC-mediated homologous recombination. Sounds technical. What's the basic idea there?
Basically, think of the BAC as a large delivery truck carrying a correct, healthy copy of the OAT gene. They used it to precisely replace the faulty gene sequence in the patient's cells with the working version.
And it worked.
It worked beautifully. OAT activity was restored back to normal levels. This proved, at least biologically in the dish, that a permanent genetic fix for this person's disease was possible.
Incredible. Okay, let's look at another RDD, a more common one maybe. Retinitis pigmentosa, RP. specifically a dominant form caused by having an extra copy of the rhodopsin gene, RHO. What did the organoids show there?
Right, a RHO copy number variation. The organoids derived from a patient with this condition showed, as you'd expect, way too much rhodopsin. The mRNA levels were about 3 log-fold higher,
Wow.
and the protein levels were massively elevated, something like 16 times more of the single RHO molecule, the monomer, and 9 times more of the paired-up version, the dimer. Just way too much protein.
Okay, so the molecular defect is clear. Overdose of RHO protein, but what did that do to the cells? What was the actual pathology?
This was the really crucial finding because it mirrored what's seen clinically. The photoreceptor cells in these patient organoids had severe maturation problems. Their outer segments were shorter. But the most striking thing was RHO protein mislocalization. Instead of the protein going neatly to the outer segment where it's needed to detect light,
Yeah.
it was getting stuck, accumulating inside the main cell body, the soma, of the photoreceptor.
And that buildup in the wrong place is toxic, right?
Yeah.
Often leads to cell death.
Exactly. That's a known disease mechanism. So the organoid successfully recreated that key destructive cellular phenotype. It gives researchers a direct target.
Right.
Stop the mislocalization. Stop the buildup.
It really validates the model for studying disease mechanisms, which leads us nicely into trying to fix these things with gene editing. You mentioned Leber congenital amaurosis, LCA7.
Yes, another dominant disease, this time caused by mutations in a gene called CRX.
And dominant means the bad copy of the gene messes things up even if there's a good copy present.
Right, it's often called a dominant negative effect. The faulty protein actively interferes with the function of the normal protein. The LCA7 organoids showed this clearly. Dysfunctional photoreceptors, problems with expressing other key genes like rhodopsin.
Okay, so how do you fix that? You can't just add a good copy.
Precisely. The strategy here was much more targeted. They used CRISPR-Cas9 gene editing, but with a twist. They aimed to specifically knock out only the mutant CRX allele, the bad copy.
While leaving the healthy copy untouched. That sounds tricky.
It is. The key was precision. They used a dual-guide CRISPR approach. They targeted the actual mutation itself on the bad allele, and also targeted a nearby, naturally occurring variation, a single nucleotide polymorphism, or SNP, that just happened to be present only on that same faulty chromosome, like a unique address marker.
So using 2 points of reference to make sure they only cut the bad one.
Exactly. This allele-specific knockout worked. In the cells where they successfully eliminated the mutant CRX, they saw really strong recovery, a big increase in important photoreceptor marker genes.
That's fantastic. It shows a clear path for treating dominant disorders by just getting rid of the troublemaker allele.
It's a very promising approach, preserving the function of the healthy copy.
So we've seen the power of organoids in vitro, in the dish for modeling and testing. But what about translating this? Is there parallel work showing therapeutic potential in vivo in a living animal?
Yes, and that's essential work too. It complements the organoid studies. There's research done in mice showing that you can actually reprogram native cells within the retina to become new photoreceptors.
Reprogramming existing cells, which ones?
Miller glia, or MG. These are normally support cells in the retina, but researchers found they could coax them into differentiating into new rod photoreceptors. It's like triggering repair from within.
Wow, how did they do that? What did they use?
They used AAVs, harmless viruses, to deliver a cocktail of specific genes transcription factors called ODX2, CIS, and NRL. These genes essentially told the Miller glia to change careers and become rods.
And did these new MG-derived rods actually develop properly?
They did. They went through the right stages, including an interesting intermediate step involving asymmetric cell division, which is typical for neural development.
Okay, but the million-dollar question, did they integrate? Did they actually work and restore any vision?
Yes, that's the exciting part. They integrated into the existing retinal circuitry. They showed electrical currents, specifically L-type calcium currents, which are characteristic of functional rod photoreceptors.
So they look like rods functionally. What about vision?
They tested that too. RGCs in the treated mice started responding to light again. And, perhaps most importantly, they recorded signals, visually evoked potentials, or VEPs, in the brain's primary visual cortex.
Meaning the signal got all the way from the new rods through the retina and into the brain.
Exactly. It confirmed that light responses were being transmitted, functionally rescuing some level of vision in these mice.
So we have the personalized testing and disease modeling power of the organoid, and proof of concept that in vivo reprogramming can generate functional new neurons. It feels like we need both approaches. But why is the organoid model still so critical for the discovery part? Why not just rely on studying actual human retinal tissue?
Well, this comes back to a really stark biological limitation of using postmortem tissue. Studies comparing gene expression in adult human retinas versus organoids found something quite alarming.
What was that?
Postmortem ischemia, basically. The lack of oxygen and blood flow after death causes massive changes in the transcriptome and the gene activity of most retinal cell types very, very quickly,
How quickly?
in less than 3 hours.
Wow, less than 3 hours. So does that mean a lot of the historical research done on donated eye tissue might have been looking at cells already altered by the process of dying?
That's the worrying implication, yes.
Yeah.
The very disease mechanisms or gene pathways researchers were trying to study might have already been significantly changed just by the tissue degrading before analysis could even begin.
That really underlines the need for a living model then.
It absolutely does. The rapid degradation highlights why having a reliable living model like the human retinal organoid isn't just convenient, it's often essential. It might be the only way to accurately study the retina's biology and disease processes before ischemia corrupts the picture.
Okay, so let's bring this all together. For someone listening, thinking about the future of treating blindness from these RDDs, what are the big takeaways from this deep dive?
I think there are two really powerful complementary strategies emerging. First, the 3D organoid platform gives us this incredible ability to model disease with patient-specific precision. Think about nailing down that 600 MME B6 dose for GA,
Right.
or validating that allele-specific CRISPR edit for dominant LCA7. It lets us perfect interventions before they go into patients.
Okay, that's strategy one, precision modeling and testing.
And strategy two is the in vivo potential, shown by reprogramming Miller glia in mice. This offers a pathway for potentially regenerating lost neurons directly within the patient's own eye using their existing cells.
It really feels like these technologies are converging, doesn't it? Being able
Yeah.
to see complex problems like the RHO protein getting stuck in RP organoids,
Uh-huh.
and then having proof of concept that you can make new functional rods in vivo, it shifts the whole paradigm. So here's a final thought for you to consider. How rapidly will things change now? Now that researchers have these tools, these living human retinal models, to test genetic fixes, screen drugs, and understand exactly what's going wrong cell by cell, patient by patient, before ever needing to start a clinical trial. That capability feels like the real deep dive advantage here.